This is for Biology 223 Genetics. I want to discuss an article published in Cell in 2017. This is specifically in contrast to the paper on long neural genes harbor recurrent DNA break clusters in neurostem progenitor cells, in which we saw they presented evidence that specifically genome mosaicism in neurotissue in invertebrates is important for brain development and function. So the idea that the diversity of gene expression mechanisms occurring in other tissues is not sufficient for the complexities of gene expression required for intelligence, and thus there's a mechanism to break DNA and rearrange it, which is pretty wild and of course comes at a great cost of potentially causing carcinogenesis, of causing brain tumors, and is rather not the way that one might prefer it to be designed. Here we have something different. Trade-off between transcriptome plasticity and genome evolution in cephalopods. Perhaps isn't obviously connected to the other paper, but let's see about that. So there's a balance between having the ability to reform and modify the totality of transcripts and the ability to evolve in cephalopods. Cephalopods, octopus, squids, nautilus, some mollusks like that. So let's read this. RNA editing. RNA editing is, for example, when we have things like C to U occur, and this is interesting because DNA, we can have, in DNA we have very commonly the deamination of cytosine occurs spontaneously at high rate. This is one of the most common DNA damages, and then of course if the cytosine is methylated, it then can turn not just to uracil, which can be easily repaired and corrected, but in the case where this is methylated for epigenetic marks, turns into thymine, and then mismatch repair can correct that. But it's a major mechanism of mutation in DNA. Here we're talking about RNA. So RNA editing, in which, and there are different forms, adenosine to inosine. There's also RNA editing that occurs in trypanosome, mitochondria with insertions, so some very strange things. The typical one in human would be C to U, and the famous case, I think the model system is apolipoprotein B, in which there's a CAA codon that is changed to a UAA codon. That's a stop. So instead of getting full length apolipoprotein B, there's a shorter transcript made, a shorter protein made, and that it has implications in lipid transport and heart disease. So that's an example. It does occur in humans. RNA editing, a post-transcriptional process, allows for the diversification of proteomes beyond the genomic blueprint. So we have genes that, we have different genes can be expressed, and then with the ability to have alternative splicing, one gene can lead to many proteins, and here we have another mechanism where RNA editing can lead to different proteins. So it's a diversification. So we have the genome can be used to make a more modified, the genome can be modified, and here the RNA can be edited such that more proteins can be made beyond the genome blueprint. However, it is infrequently used among animals, metazoans in general, for this purpose, including us. Recent reports suggesting increased levels of RNA editing in squids thus raise the question of the nature and effects of these events. So the question then that they're asking is, if squids undergo a lot of RNA editing, what's going on there and why? And squids are interesting because squids are behaviorally sophisticated, they're intelligent. We show here that RNA editing is particularly common among behaviorally sophisticated colloid cephalopods. So they mean smart ones. So common, it's not rare in humans, it's quite rare, with tens of thousands of evolutionarily conserved sites. So they have identified in the genome many sites. That's one important thing. It's heavily used, RNA editing, and it's evolutionarily conserved within these behaviorally sophisticated cephalopods, which implies that it must be very important or it wouldn't be conserved. Editing is enriched in the nervous system, so that's where it gets really interesting. Why in the nervous system? What is it about the nervous system that requires this special extra mechanism in order to create a diverse proteome? We might say, well, that's kind of strange, but if we compare it to the example in mammals of the genomic mosaicism in brains, and then I think, okay, something about intelligence requires some extra mechanisms, just like the immune system for antibody diversity. So editing is enriched in the nervous system, affecting molecules pertinent for excitability and neuronal morphology. The genome, so that makes sense, and it's about their function. The genomic sequence flanking editing sites is highly conserved. So this is a phenomenon that they're highly conserved. Where editing occurs, you might say, well, if this editing occurs in apolipoprotein, for example, it doesn't occur at every cytosine. It occurs a specific one, and there's an RNA sequence there, and there's mechanisms by which are easy to conceive of where there's an RNA structure or a binding site for a protein. Okay, so the sequence flanking a site that's edited is highly conserved raises two possibilities. There's a direct RNA sequence binding protein, or there's some RNA structure that forms like a hairpin loop, which is recognized by a protein. The genomic sequence is highly conserved, suggesting the process confers a selective advantage. If it didn't confer a selective advantage, if it was by chance, it would not be conserved. So and notice they say suggest. Due to the large number of sites, it's not just one site, the surrounding conservation greatly reduces the number of mutations in genomic polymorphism in protein coding regions. So what they mean is that so much of the protein is conserved because of these flanking sequence that the proteins cannot mutate. There's not as many neutral mutations possible. The proteins, a sequence can mutate presumably very easily to go to synonymous codons. So we can expect that protein coding sequences can mutate relatively easy by altering their codons to a synonymous codon or a relatively neutral mutation and by change, a conservative change. But this has so much of that, there's so many of these sites that the selective advantage conferred by having these editing sites limits the ability of the proteins to mutate. And that has some important theoretical considerations that are a bit beyond the scope here, at least the scope of this course. The surrounding conservation greatly reduces the number of mutations, genomic polymorphism, in protein coding regions. So they're harder for them to mutate, harder for them to evolve, at least under purifying selection and drift. This trade-off between genomes, so this trade-off that there's this extra-regulatory mechanism that's a selective advantage, this trade-off between genome evolution and transcriptome plasticity highlights the importance of RNA recoding as a strategy for diversifying proteins, particularly those associated with neural functions. Okay, so they are reporting this phenomenon, which is very interesting in itself. It has a ramification that the this plasticity that occurs by the RNA editing comes at the expense of the genome having restricted evolution. This is related also to the concept of the benefits of sexual reproduction versus asexual. Some deep theoretical issues here, possibly. And it highlights the importance of RNA recoding, the strategy of diversifying proteins, which we hadn't really considered before because it is pretty rare in animals, and in humans it just occurs in a couple, some places. So it's not something that people seem to have thought much about, and particularly those associated with neurofunction. And then this is reminiscent of the paper on the recurrent double-strand break clusters and neural cells, as something is special about neurofunction, something special about the nervous system that requires extra mechanisms of acquiring a diverse and regulated set of proteins. So intelligence seems to be dependent upon extra levels of regulations and extra levels of protein diversification. I will stop here.